Talking about a project that is actually a very close collaboration with Nadia Tarasova, the next speaker. So in reality, it's like one extended presentation between the two of us. And you will see here and there some duplication of slides. And I will presage some of the stuff that Nadia will be talking about. So our ultra-large library SAVI, which stands for Synthetically Accessible Virtual Inventory, came from the idea that why are we even fighting with the question, how do I synthesize a molecule? There are 10 to the many molecules in chemical space. Some people say 10 to the, 34, 10 to the 60. Dave Weininger said 10 to the 200. It's many. And if we assume that a small percentage of them are easy to synthesize, you end up with something that is still huge, 10 to the many, okay, like 10 to the 32. And if we design in the computer a subset of these many, many molecules that are easy to synthesize, then we're kind of done, right? We just apply computer-aided drug design approaches, docking, pharmacophore searches, and we know that these molecules can be made. So this was the idea behind SAVI, which the idea started roughly 10 years ago, okay? So what do you need for something like that? You need building blocks, and we have been using for quite a few years the building blocks from Enamine in Ukraine, and Yuri Maroz has been very helpful in making those available to us. You need rules how to put these building blocks together, transforms, as we call them. And this is kind of a bit different from what most other projects that are generating ultra-large libraries do. And I mentioned mostly Phil Judson here at the company LASA, who has been writing such rules. And I will tell you a little bit how we got to it. And then you need a chemoinformatics engine that puts these rules together to form new molecules. And that has been the chemoinformatics toolkit Cactus, written by Wolf-Dietrich Ihlenfeld in Germany. And in my group, we have combined all these components and have made new molecules, and quite a few of them. So the transforms, what we did, we actually went back through the past to the future, if you will. So this is a set of rules that is basically an export system. It is a language pair called Chemtran and Patran that was the basis of the LASA project. If you will, the computational embodiment of E.J. Corey's Nobel Prize winning work on retrosynthesis. But we use it forward synthetically. These rules go way beyond what in most cases people use for making more new molecules chemoinformatically, which are SMERX transforms. Right? So this LASA approach, for example, has a scoring system built in. So you can add and subtract scoring values if the logic says, well, this synthesis may not work too well, or the yield may be lower. And very crucially, you can have kill statements that the logic says, this, forget about it. Don't do it. So we got access to original knowledge base, which really started 50 years ago. So that's why Chemtran and Patran is a very strange language. It's like synthetic knowledge, basically written in an ancient Fortran style with a lot of buzzwords in it. But I'm not going to go into the details of it. But the development of these transforms petered out in the 1990s. And so important stuff was not in there, such as Suzuki coupling. So we have been writing new transforms in this language. We have intentionally focused on coupling reactions because we want to make interesting new molecules. We have not done much function group interchanges, although that's all possible in this approach. 16 of these transforms are actually ring forming because we wanted to form rings and interesting rings and get also, if possible, out of flatland. So the total number of transforms used for what we call SAVI 2020 was 53. So there's 13 that came from the original knowledge base, and we wrote 40 new ones. So the building blocks, this is what you need to actually build something that can be synthesized. So that was one of the main reasons to go to Enamine. There's other building block sets that you can look at, but Enamine actually makes these compounds for you if you pay them for it. And that's crucial for the project, because we don't have hundreds of synthetic chemists at NCI who just wait to synthesize molecules. So we got, for SAVI 2020, about 150,000 building blocks, for which at least 1 gram was available. And then you have to match them with the required reactive groups in these 53 transforms. And so about 143,000 matched at least one of the transforms. Very crucially, all the reactions that we ran in and for SAVI 2020 are single-step syntheses, the simplest syntheses that you can think of. A plus B gives C. Okay, so the process is reasonably complicated, so I cannot go into the details, but since in principle the LASA approach is still retrosynthetically, so what we do, we build a proposed product, and then we ask the transform, can the proposed product actually be synthesized with this transform? And only if the transform logic says, yes, it is possible, then we keep that. So we had an initial set of about 3.6 billion reactant pairs, and a good half of them were killed. And that is desirable because we want to reduce the haystack. Right? We want to make only molecules that we know can be synthesized. So we ended up with about 1.75 billion accepted reactions. Now, as I already mentioned, we have scoring in these rules. So we can actually subdivide the total set of generated molecules into different classes. And we've done that, and we call the plus class, which was about 1 billion molecules, what had those molecules that didn't see a single subtract, because you can have multiple, and you can have subtracts and adds, you know, as you go through the logic, okay? And then you have neg classes that have had more and more severe subtract, scores or demerits applied. Okay, and that gave a total of about 1.75. And there is a certain duplication, which is good. So some of the molecules can be synthesized by several routes, and there's nothing wrong about it. So the whole data set is actually fully publicly available. You can download it. It's terabytes of structures. We have it in SD, and we have it also a little bit more compact as SMILES tables. So currently if you can, if you want to, you download it, and then it's your problem what you do with it, right? And Nadia will briefly mention something that we're working on to make it interactively searchable, but that is not entirely trivial if you are looking at billions of molecules. So these are not just molecules that are some weird structures, you know, we wanted to make molecules that are useful for drug development. So we looked at a whole bunch of properties that are looked at, and this is just a subset. You know, if you want to see more, you should look at our paper, but they look really kind of nicely. For example, if you look at the flatness, the FSP3 value, you know, you see that we are not totally in flatland. So we have molecules that are not just flat planks, and this is of interest nowadays, right? Similarly, the drug likeness score, QED score, is actually even better than, you know, from the original paper, some of the drug molecule databases that were analyzed. So this all looks pretty good. So we said ourselves, okay, these molecules look reasonable that we made. We also think that we have created quite a significant number of novel ring systems. I say think because it's difficult. You need to compare with existing ring systems. And we have done some manual searches in SciFinder, but what we would need to be able to do is download all of the CAS registry. And as you know, that's not really possible, right, to do the overlap analysis. But we estimate that we have generated at least 1,000 novel ring systems in these 1.7 billion. Okay. An interesting test for us, or analysis, was to calculate the overlap of SAVI with other databases. And at the top, the real database, which is the database that many of you will know, which comes from Enamine, is quasi the sister of SAVI, because it's essentially built on the same building blocks. And there is a certain overlap, but it's in the range of 8 to 10%. And that was a very good result, because it showed us we're not generating weird stuff, you know, but we're still generating mostly novel molecules, because the rules that Enamine uses, the chemistries to make real, are quite different from what we have in SAVI. And we actually exchanged the chemistries so that we could see, and that we wouldn't accidentally try to duplicate what they're already doing. And that has been quite successful. And the overlap with other large databases, such as the iResearch Lab, which is an aggregated database of screening sample collections from ChemNavigator, which was then purchased by Sigma Aldrich, is only below 1%. And that's typical, that you see some overlap, but actually quite little. So you make mostly novel molecules in these approaches, and that's good. Okay, but the molecules should not just be in the computer. The molecule should be actually synthesizable and be synthesized. And in the project that we know of, we have synthesized or had synthesized about 170 molecules in anti-cancer, anti-inflammatory, antiviral projects. And they all came from the plus subset, because that's already billion, so that's kind of enough. Most of them were synthesized by Enamine, and the success rate of these syntheses was 97%. So that was astounding even to ourselves. You know, I would have been happy with 80%, but practically all the predicted syntheses worked as proposed by the SAVI approach. Now, the transforms, and I don't have the time, and Nadia will talk a little bit more about that, there is a connection between what transform you use and molecules from which transforms you analyze and obtain in docking and other CAD approaches, and the binding site, where you actually want to inhibit or activate something, bind something. So for example, here the Sonogashira coupling creates a rigid scaffold, and that's good for some binding sites. So there is a connection of what the transform does and can do, and what you may find in terms of active molecules. Also, you see cases that parts of molecules that are generated by the transform in the reaction, you know, may contribute directly to binding. And that tells you that the pure fragment-based approach, when you simply take the blocks, may not tell you the full story of what's active. So that's something to keep in mind. Okay, so one of the questions that people had several years ago, does it even work? You know, does this virtual screening with large or ultra large databases work? And we would say yes. Now it works. You know, you have docking software that is really good, and we are obviously here to discuss some of the software. We have sufficient computation resources. You have computer clusters, you have cloud computing, you have GPUs, as was already mentioned. We still would like to have more, but at least you can do a lot with what you have available. The protein structures are getting more numerous, that's for sure. They're getting better, but there's a big but, and Nadia will be talking about this. And of course, you have all the approaches such as AlphaFold to calculate such structures. So that's exciting, but I think we're at the very beginning of useful results from these efforts. And then, of course, you have large virtual libraries of synthesizable compounds, and SAVI is one of them. Of course, virtual screening for well-defined pocket has been around for a while, but people said, well, but you cannot, target so-called undruggable proteins, cleftless proteins. Forget about it. It doesn't work. And the question that Nadia and I have been discussing in our groups is, is that really true? Or have we been searching in the wrong part or too limited a chunk of the chemical universe? And then the next question is, well, do we have the data and tools to venture into these spaces? Right. So data would be, ultra large databases, and the tools. Well, this is what we've been using, and some of these tools come from Molsoft. So up until 2021, roughly, end of 2021, early last year, we had a typical screening workflow that was based on ICM Pro docking. So first, we needed to verify the X-ray structure suitability for the virtual screen, and that is something that Nadia will be talking about. And it's not unimportant, okay? And then we docked diversity sets, a small subset of both SAVI and enamine real, because we can actually order compounds from these two databases combined in one order to send to enamine. So we usually check both of them. Because the docking with a CPU based ICM Pro software, even with the cluster that we have at NIH, would take nearly a year for all of SAVI. So that's not realistic. So we take a small subset of about 3 million molecules. We pick the best scoring compounds. We have them synthesized, we test them. And then there is fragment-based, or there was, I should say, fragment-based and 3D similarity searching of the entire database, which you can do much faster. And then you redock the analogs with ICM Pro. And so you have this cycle of drug development. And that has been quite successful. And I will only briefly report one project that was, again, scientific. In terms of the wet chemistry, everything was done by Nadia's group, so my group is completely computational. We do nothing wet. So this is a STAT3 N-terminal domain. This is transcription factor, and it has been found to be of interest both in cancer as well in antibacterial context. For example, Nadia and collaborators, they showed that if you inhibit STAT3, you can kind of essentially cure mice of tuberculosis, right? So it turns out that the STAT3 N-terminal domain has sort of a pocket, not a deep pocket, not like a glove, of more traditional targets, but something you can put something in, and so these were compounds from both SAVI and real that were docked into this crystal structure, and the best binding results were shown to actually directly interact, by microscale thermophoresis. And they were also toxic to mouse pancreatic tumor cells. And this approach actually allowed us to file a patent application. And the whole process from the initial in silico screening to submitting that patent application with the pandemic in between and shutdown of labs and everything was 15 months. So I think this shows that these approaches are really powerful and allow one to move forward quite fast. Now, what can we do now? The RIDE program, Rapid Isosteric Discovery Engine, was already mentioned, and we have been using this for like a little bit more than a year now in, I would say, collaboration with Eugene. you know, getting updates and sometimes saying, well, this doesn't really fully work, and can you improve that, please? So RIDE, as was already mentioned, uses pre-generated databases of conformers, so-called MOLT files, to do ligand-based search. This generation of conformers is pretty expensive. We spent for SAVI about 8 million CPU hours to generate it. But you do this only once, then you have it. And then if you run RIDE on GPUs, it can run on CPUs, but if you run it on GPUs, it is much, much faster. Especially if you read these data from SSDs. So what you essentially create, and you will see some performance numbers in next slide, you create a gigantic GPU, you know, with terabytes and terabytes of space, and that is really super powerful for this code. So the new screening workflow now is, so we do the initial docking of the diversity set as before, after the preliminary steps even, and Nadia will explain this in some more detail. So this is structure-based screening, okay? Then the best scoring hits are synthesized and they're assayed. And then if there's actives among them, then we do write screening of the entire database, or entire databases, you know, SAVI, real, and meanwhile others, with the active. So then we switch to ligand-based screening, and then we take the top hits from the write screen and dock them again with ICM Pro, okay? And that again is the new 2 or even 3-layered cycle, if you will. So what have been our experiences with RIDE? So
hey Mark, there's 5 minutes left.
5 minutes, yep, this is last slide, so yeah, we're gonna be fine. We are using 2 pretty powerful graphics workstations because we need the GPUs. Okay, they come from the company Exact in California, and we have one that has 4 NVIDIA RTX 2080 workstations, and one newer one that has 4 NVIDIA A6000 GPUs. Okay, so it is important that you split the ULDB in several chunks because you need to send the chunks separately to the GPUs, okay? So for SAVI, we split it in 13, which was sort of, you know, taking the transforms and distributing the products roughly equally according to the transforms, and which I didn't mention, but the transforms that we've used with the building blocks that we have generated very, very different sets of product results. For some transforms, it was only a few hundred molecules. For others, it was 500 million, okay? So, you know, you need to play around with that. And then what you should do for the classical, no, I mean, the version of RIDE as it was available up until a few months ago, you know, you should have it on fast SSDs. So you distribute these chunks as evenly as possible across, in this case, 4 large SSDs, because you have 4 GPUs, right? So large, I mean 8 terabyte or 16 terabyte. Not super cheap, but it's worthwhile doing, okay? And then you run 4 jobs in parallel, each one reading from 1 SSD. And there's total wall time then is when the sets of jobs say, you know, we have 13 chunks, so you have 3, 3, 3, and 4 jobs that are run sequentially on each of the GPUs, but for the 4 GPUs, they run in parallel. That's the wall time you get out of it when the last of these 4 shell scripts essentially has finished and all the jobs have terminated. Okay. So the write queries can use, and typically we do use, a single structure, you know, maybe the most active molecule, but you can put several structures into them. So we tried up to 10 That was the max that fit into the memory of the GPUs. And we've run quite a few jobs with 6. So that gets quite a bit slower, not necessarily linearly, but it slows down. Okay. The total number of hits strongly depends on a cutoff parameter for this atomic property field. So typically what we want to have for redocking is a few 100,000 to maximally a few million out of the billion. So you have to play around. Actually, it's what I typically do is I run a little test job, that I explore different values of the APF cutoff parameter on the diversity set. That takes only a few minutes. And then I say, okay, 0.6 is good for this target and 0.4 for another. So this is very target dependent, and you need to test it for each target. So the wall time to query all of the 1 billion plus subset with the 2080 GPUs was around 7 hours. So I can now screen all of SAVI that interests us, in 7 hours versus like 1 year for docking. Okay. If I run multi-compound queries, the longest wall time was about 35 hours. Okay. An interesting and useful parameter to look at is the conformers per second. And that depends also strongly on what molecules you have, what small molecules in the ultra-large database you have. Of course, it depends to some extent on the power of the GPU, but maybe less than you might think, So the typical values we see is between 200 and 500,000 conformers per second screened. We have seen up to 720,000 conformers per second, but we have also seen numbers below 20K, if you have multi-compound queries. So it's a range, and there is a good reason why you have this range. Now, quite recently, a write has been made available with being able to read compressed mold files. And that has advantages both in terms of the space you need for these files, as well as some speed advantages. So with the faster GPUs, we've now seen that SAVI can be screened in like 2.5 hours, to 9 hours. And there we have seen up to over 800,000 conformers per second with a real database. So in general, we've actually seen that the real compounds seem to be a little bit smaller and a little bit less flexible. So you have fewer conformers. We have not systematically analyzed that, but that's our assumption, why real generally is a little bit faster in these write screens. And with that, I'm at the end, and I would just like to acknowledge a lot of people. I've not talked about a lot of parts and aspects and the history of SAVI. So I just leave this screen up here, and I'd be happy to answer any questions.